Hey guys, today's video is going to be on muscle contraction, and I'm going to cover basically all three major muscle types because I thought it'd be better to get a holistic picture of what's going on. So I'm going to be going through skeletal muscle, cardiac muscle, and smooth muscle, how these muscles get to their contraction phase or cross-bridge cycling phase, and how the cross-bridge cycling is stopped. As well as I'll be going through some key anatomical features of each of these muscle types. So skeletal muscle we've covered already. We have our action potential coming down from our brain to our ventral horn, then our ventral horn sending out motor neuron axon to our muscle where acetylcholine is released. Then the acetylcholine depolarizes the muscle cell, and this depolarization travels down the T-tubule where dihydropyridine is depolarized and changes its conformation, and this dihydropyridine is linked to a ryanodine receptor or an RYR1 receptor, and the RYR1 then changes its conformation and allows calcium to flow out of the sarcoplasmic reticulum. So here we have our calcium flowing out of our SR. Calcium binds troponin C. This moves tropomyosin out of the way, lets myosin bind, and lets our cross-bridge cycling begin. And then finally, when we want our muscles to stop contracting, we have something called a SERCA pump, which is a sarcoendoplasmic reticulum calcium ATPase, which is basically a calcium ATPase that pumps calcium back in to our sarcoplasmic reticulum using ATP. Okay? So let's move on and talk about a different muscle type. In this case, cardiac muscle. So cardiac muscle is also a type of striated muscle, and we're going to be comparing it mainly to skeletal muscle when we talk about the details of how it gets activated. So here, instead of our brain actively thinking about moving the muscle, because you don't actively thinking about pumping your heart, so we have something called pacemaker cells, which consists of your SA node in your right atrium and the bundle of His and Purkinje fibers and so on. These basically do the same depolarization step that your motor neuron did on your skeletal muscle. So they depolarize your cardiac muscle, and this depolarization travels down the T-tubule the exact same way as happened in the skeletal muscle. The T-tubule then depolarizes, and the dihydropyridine receptor there changes conformation. Now this dihydropyridine receptor is different than the one found in skeletal muscle because unlike skeletal muscle, it's not linked to ryanodine receptors. So here we have our T-tubule and our dihydropyridine receptor, and this dihydropyridine receptor in our cardiac muscle acts as a calcium channel. So it's going to act as an L-type calcium channel and let calcium from the outside enter in the cell. So this calcium then does its action by going to the sarcoplasmic reticulum, which I've drawn here as SR, and it changes the conformation of the ryanodine receptor, which then allows more calcium to flow out and do its action. So here, the dihydropyridine and ryanodine are linked, and their changes in conformation are also linked, which directly affects the sarcoplasmic reticulum. And in cardiac muscle, the dihydropyridine receptor acts as a calcium channel, and this lets extracellular calcium come in and act on the ryanodine receptor, which in this case is a ryanodine receptor type 2, here it's a type 1, which will do the same thing that the ryanodine 1 does. It will let calcium flow out of the SR, then the exact same thing happens as in skeletal muscle. Troponin C binds calcium, moves tropomyosin out of the way, then we have cross-bridge cycling. Okay, so we've covered cardiac and skeletal muscle. Let's talk a little bit more about their differences. So in cardiac muscle, we have something called calcium-induced calcium release because the extracellular calcium is what causes intracellular calcium release. Here in skeletal muscle, we have extracellular depolarization causing intracellular calcium release. So let's say we dropped each of these muscle types in a vat full of calcium. Skeletal muscle would not contract because all of the calcium that's required for contraction is stored on the inside and released via depolarization. Cardiac muscle, on the other hand, if the concentration of calcium is higher on the outside than on the inside, calcium could theoretically flow inside the cardiac cell and activate depolarization, which would then cause more calcium release, so depolarizing the cell and activating the ranitidine receptor. So if you dump cardiac muscle into a vat full of calcium, it might contract. Next, we have our diagram over here, which I think is important to talk about just briefly. So here we have one cell, so this is a electron micrograph of just one cell, and you'll notice these structures, and these are structures commonly associated with striated muscles. So at the end here, which I've highlighted in red, we have our Z lines, and in the middle we have an M line, and I'll just outline some more of the key structures here just so you can get a quick introduction to them. So on either side of our M line we have an H band. So this H band consists of only myosin. Then at this thick darker section is called an A band, and this consists of the entire myosin fiber. So myosin is thicker than actin, so you'll see a darker region. That's our A band. And at the ends here we have light bands. These are called I bands, and because actin is thinner than myosin, these I bands consist of only actin. And what's important to note here is this structure right here in the AI junction, so between the A and the I bands. These are called triads, and what they are is basically one T-tubule, which you'll find here in the center, surrounded on either side by a terminal cisternae. And what terminal cisternae are is basically the edges of a sarcoplasmic reticulum. So our T-tubule needs to dive into our muscle structure, as I've talked about in the skeletal muscle video, and needs to activate our sarcoplasmic reticulum. So here we have one on either end. This triad structure is not found in cardiac muscle. So in cardiac muscle we have the same striation structures, but instead we have something called a dyad, where we have only one T-tubule and one terminal cisternae. So that's an important electron micrograph finding if you're ever asked to differentiate between the two. You have dyads in cardiac muscle and triads in skeletal muscle. Let's talk about our final muscle type, which is smooth muscle. So smooth muscle has a more different way of being activated than cardiac and skeletal muscle because its structure, as you can see in the top right here, is quite different than this striated form. So it is not striated. It is also under autonomic control. So cardiac has pacemakers, skeletal is under somatic control, and smooth muscle is under autonomic control. So that means you don't have to actively think about it for it to move. So this is, for example of this is the muscles in your gut, so they're involved in peristalsis, or any kinds of movements in your body that you don't have to actively think about. These are smooth muscles, and how they're structured is these yellow bodies over here, these little yellow spots, these are known as dense bodies, and between them you have intermediate filaments, which are outlined in here in blue, and also between them are actin and myosin filaments. So these are the darker structures right here. So what intermediate filaments are is something called vimentin and vimentin-like structures, and they essentially establish this web to help the muscle keep its structure. And these stripes molecules over here that you'll see, these are actually the contractile units. So they are the sarcomeres, which you're going to see similar to these sarcomeres down here. But you won't see the striated structure because they are irregularly placed. And when they contract, they make the muscle shorter. So when muscles contract, they get shorter. That's similar for all three muscle types. So how do you activate a smooth muscle? Well, smooth muscles on their surface have a calcium-gated channel. And this channel can be activated by multiple types of stimulus. So you can have stretch, you can have a ligand gate, you can have a voltage gate. It really depends on where the smooth muscle is and what are its properties. But you will have a calcium-gated channel, and then calcium will enter the cell and will bind a molecule called calmodulin. So calcium and calmodulin will form a complex. This calcium-calmodulin complex then goes on to activate something called myosin light chain kinase, which will be very important for activating our myosin. In smooth muscle, myosin needs to be phosphorylated to be activated and undergo cross-burst cycling. So our kinase over here is going to phosphorylate our myosin head, which will let it bind actin and activate cross-bridge cycling. Now remember, in our striated muscle types over here, that our actin was our inactive molecule. So our actin needed tropomyosin to be moved out of the way in order for cross-bridge cycling to occur. Here our myosin is the inactive molecule, and it gets phosphorylated to activate cross-bridge cycling. So once cross-bridge cycling stops, just like in striated muscle, we need some way to stop it. So the molecule that does this is called myosin phosphatase, which does the opposite of myosin light chain kinase. It's going to remove that phosphate group off of the myosin head, and it's going to let the contraction slow down and stop. So myosin phosphatase and MLCK are in competition, and depending on whether extracellular calcium is coming into the cell will determine which one is predominating, whether the muscle is contracting or relaxing. And one final important thing I would like to talk about, which I should have mentioned earlier, is cardiac muscle structure, which I've outlined down here. And basically, cardiac muscle cells are attached to each other using a couple different important attachment molecules. So we have apical attachments, so stacked one on top of each other, and here we have lateral attachments. For apical attachments, we have things called desmosomes and gap junctions, and then for lateral attachments, we have desmosomes and adherence junctions, which are also known as fascia adherents. And what these lateral attachments do is they hold the heart muscle cells together to prevent them from separating during contraction and expansion. So if you can imagine the heart beating and it expands and contracts, these attachments prevent the muscle cells from separating. What the apical attachments do, besides holding the cells together, here are our desmosomes do that, they also transmit the calcium signals between cells. So let's say our Purkinje fiber activated this cell down here. Well, we want the activation potential to move between cells. And these apical attachments, or these gap junctions, are going to let calcium from this cardiac cell move into the next one and activate it. So that's very important. So our action potentials move apically, or also basally in this case, so any apical or basal attachments, between the cells via these gap junctions. Skeletal muscles don't have the same feature. Alright, well, I'll see you guys in the next video, and I hope this video helped.